Hi, everybody. I'm Jen Dawson. I'm behind the camera today as I'm a bit under the weather, but I'm so thankful that Dr. Amy Powler has joined me for her take on the meeting so far. If you don't know, Dr. Powler is the chair of the Department of Dermatology and the Walter J. Hamlin Professor of Dermatology at Northwestern Feinberg School of Medicine. She is also one of the founding members of PEDRA. It's great to see you here, Dr. Powler. There's a lot to digest, a lot to talk about. So you can give us like your top highlights and then feel free to go into any detail.
There's so much to talk about at this meeting here in Japan. It's been so great to see international colleagues that we haven't seen for years here in Tokyo. And of course, there's a lot of very exciting science. I don't even know where to get started in terms of pediatric dermatology. I think I'm going to talk a little bit about mosaicism.
That sounds great.
There were two presentations that I thought were quite interesting to pediatric dermatologists. One of them is given by Howard Chang from Stanford, and of course Howard is brilliant and is always working on something that is so illuminating. And his talk today was not an abstract presentation, but an invited talk about some of his recent work with the X chromosome, and particularly a gene called ISID or XIST, depending on how you might pronounce it. It's spelled XIST. And we've known about XIST for a long time, that it is what on the X chromosome dictates that chromosome will be silent. And his group has gone on to study that a bit more and to ask some very important questions about it that I think most pediatric dermatologists may not realize. So I got a little bit of notes here, so I'm going to look at them every so often.
Please share, yes.
Okay, great. So really exciting work. We've known that this gene ISID is critical for X chromosome inactivation, and again that there's one copy per cell that is very involved in females in turning off and allowing the other gene, the other chromosomes, X chromosome, to be the dominant one. So it's essentially an epigenetic regulation, as he called it. There's epigenetic memory, because what happens very early on, and if I recall, 12 to 16 days gestational age in females.
Wow.
Your decision is made forever in each, for example, stripe of flesh goes lines for epidermis, and certainly this occurs in every cell. So we really need to recognize that these kinds of patternings are decided early on. So epigenetic memory, and it lasts for a lifetime, and we essentially have shut down in the reading and translation of genes in that particular chromosome. So he's been able to show how the active ISID just basically condenses the one group of chromosomes into basically a bar body for that X chromosome in those particular cells. But he asked a bunch of interesting questions, and here's where it gets kind of cool. So he first asked, what are the protein partners of ISID? Because when you identify protein partners, you can start to ask, what are the interfaces? What are the interactions? How is ISID working? And he identified 81 protein binding partners, direct or indirect. And he also noted that this particular gene is a very large gene that has a lot of repeats. And there's one particular repeat called an A repeat that's at the 5' end that's really been important for gene silencing. And looked at proteins that were near that region that seemed to be unique, and found one called SPEN, S-P-E-N, which seems to now be the key silencing factor based on his work. Highly conserved, it's in fruit flies, lots and lots of very primitive organisms. So we're talking about something that's essential and has been there a long time. Now, what he looked at was, what does it look like in that particular configuration, in that 5' end repeats? And he found actually that it very much resembles repeats that are seen in retroviruses. And ultimately decided that what's happening here is viral mimicry. And so it's not just that we're talking about a role in turning off a chromosome, but this gene has an element in it that basically helps to protect against viruses by dialing down, making it kind of quiet and not being producing, not translating, not making proteins when there is something around that seems to be like a virus. Next question is it just something that gets turned off early on and stays? How does it stay? Why is it that particularly cells that have a lot of turnover, a B cell, a myeloid cell, forever has that one X chromosome turned off? It must be very important there. And it turns out there are mechanisms that just keep it persistent and still to be discovered. But it's not just a one-time thing. It's a persistent, active, turn off, active, this exists gene. Now, the other thing in looking at all of these protein partners and all, he found out that 22 of the top 45 are proteins that are targeted for autoimmune diseases. Example, Rho, La. We are familiar with these from lupus, for example, and Sjogren's syndrome. These various autoimmune diseases, as we all know, are much more common in females than they are in males. So he then asked the question, could somehow this ribonuclear protein complex that includes exist be a trigger of autoimmunity? Lots of repeats, long gene, could be prone to polymer cross-linking proteins that could activate B cells. And in fact, make naive B cells become atypical cells, like what you see in lupus, rheumatoid arthritis, in COVID-19, which we know is a disease where there was a lot of interferon activity. So he created mice in which he basically took male mice and introduced the exist gene. Now you can't just do that, right? He did it in such a way that he could control it by making it inducible. It was inducible with doxycycline. He gave doxycycline, he could induce it. So he'd have female mice as a positive control. He'd have a bunch of negative controls. And then he'd have these male mice in which he can control the activation of that extra exist.
Wow.
And he did it on a different chromosome, not the X chromosome. Right. So, and what he found is that in these male mice, they created bar bodies with this introduced exist. And he also then found that when he took a technique that's been used to cause lupus, which is giving them something called pristane, that, of course, the females had a high propensity to develop lupus, including lupus for organs. The negative controls, not so much. These males, though, with the exist, when induced, were very similar to what we're seeing with females. So some real evidence of increased autoimmune antibody, increased organ involvement, like renal disease, like is seen in lupus, just from introducing into the males this exists. And that suggests then that there might be some role for this exist gene as in autoimmune disease in causing it. And of course, mechanistic studies have shown an increase in toll-like receptors in terms of the pathophysiology of these diseases turning on, kind of viral activation type pathways. And so there might be a similar type of mechanism. And finally, he went to patients and took patients with autoimmune disease, looked at their sera, and found a whole bunch of patterns that involve these types of genes that overlap or are distinct for particular subsets, but nevertheless seems to be some early evidence that this might be true in humans as well. So I think that was fascinating.
Yeah.
It was very new to me that as someone who's very interested in mosaic disease and has long been interested in the mosaicism that we females have in terms of why we have this exist gene and why we have one active, one inactive that's forever. And so I think this is just the tip of the iceberg in this brilliant scientist telling us about that. I want to mention one other. I know I've been rambling for a while.
No, that's all right.
Of course, Veronica Kinsler is always someone who adds to our knowledge base in pediatric dermatology. She's here at the meeting, and I was absolutely fascinated by some work that she has just started, where she's been thinking about the GNAC and the GNA11 mutations that occur, let's say, in Sturge-Weber syndrome, and also the fact that there's an alteration in MEK pathway signaling, but MEK inhibitors haven't really knocked our socks off for these diseases like Sturge-Weber with the capillary malformations, or frankly, other cells that they might be in. So she thought about it and thought about the tram track calcifications that we see in the brain in Sturge-Weber syndrome, which is one of the hallmarks that goes on with that particular disease, and wondered whether there might be some abnormality in calcium signaling in these diseases that might be a primary defect that could be targetable. So what she did, created some endothelial cells. She mutated them by using the GNAC R183Q, the hotspot mutation that's been described with patients. And she also had other cells that she introduced mutations in both GNAC and GNA11. And what she found is that through the introduction of these mutations, there's an increase in intracellular calcium signaling, and also more prolonged intracellular calcium signaling. And this is through certain channels that are known to be involved in calcium signaling called crack channels. She said, well, what happens if we just take calcium out of the medium and then compare them to my control cells that have been mock altered or unaltered? And once you take calcium out, I assume by EDTA, they were the same. So this alteration is only seen when there's added calcium to the medium. She then thought about, well, is this relevant to patients? And she went to the literature and found that there is evidence that there is reduced ionized calcium in a fair number. She cited 41% of patients, and especially those with intracranial calcifications, especially an association of this reduction in serum ionized calcium associated with seizures, again suggesting that there may be some tracking of calcium intracellularly into organs like the brain. And so she thought, well, gosh, maybe there's something to this, and took crack channel inhibitors. Now, these are not used in dermatology, but crack channel inhibitors are used in oncology. And she was able to treat these mutant endothelial cells with the crack channel inhibitors, and it corrected it.
Wow.
So she is planning a study with patients, presumably with Sturge-Weber, with some of these crack channel inhibitors, and stay tuned. But I thought that that was also another mosaic disorder with some wonderful pearls from Veronica Kinsler from London. So maybe I'll stop with that.
That was amazing. Thank you so much, Dr. Paller, for those highlights. It sounds like those talks were really incredible and really transformative. It sounds like they're really
I think there's some pretty exciting breakthroughs here. And we're not even halfway through the meeting.
Right.
So lots more to come.
Yes. Thank you so much, Dr. Paller.
Great to talk with you, Jen.
Hello again, everybody. I'm back behind the cameras. I'm feeling a little under the weather, but I am delighted to be joined by Dr. Reed Oldenberg. He is an assistant professor of dermatology at UCSD, and he's also serving on PEDRA's Early Investigator Committee. And I've been bumping into you throughout the meeting. You've been having a great time. So what are your highlights? What are your big takeaways?
Right. I mean, it's been a really fun meeting so far. I'm certainly finding myself running from room to room, like trying to keep up with all this stuff. I mean, the list is just loaded with excellent speakers. Everything's been very inspiring, but I think I can highlight a couple of talks. I mean, one of them definitely was Chris Nagao from the NIH, just a fantastic talk where he really showcased essentially, I think a decade to 15 years of research going through identifying different cell types in the skin. And then it got a little bit more high level. And I think for me, what really resonated with me was the study that they had done looking at using single cell RNA sequencing from a DRESS patient that was very difficult to treat. And by looking at the different profile of the cells, both in the skin and the blood, in a patient that had failed multiple different immunosuppressive therapies, they identified a signature that was with JAK stat elevation, and then they ended up actually treating that patient with JAK inhibitors. And a lot of that really inspired me to focus my research on identifying inflammatory pathways that are dysregulated. So that was really cool to see him showcase that. And then he has since, and I think this is sometimes the advantage of having close collaboration with clinicians and having a big network. And he has since expanded his research to include investigating Degas disease and using similar platforms to look at Degas disease, which what we always learned about was related to potentially a vasculopathy or a vasculitis picture, but it turns out it's once again related to an inflammatory dysregulation where the T cells are acting up, specifically CD8 T cells, and the way he spoke about it was really inspiring because and I think because he's an expert clinician as well, he started with the histopathology, so it felt very much like residency, looking actually at the tissue, and very astutely identified that there was an interface reaction pattern. So there's a lot of lymphocytes, and then actually taking that into the single cell matrix and looking at what those cells were, finding the signature, and then bringing it back and confirming it using immunohistochemistry. So that was really great for me because as a aspiring physician scientist, that's ultimately what we want to do. We want to take things from the high level of what is happening to the patient and try and understand it better with the idea of improving treatment options. And I think that's really what this is all about, is trying to use the best that science can offer to improve treatment down the line.
And that really comes through when you're talking, just in the times that we've had a minute to chat, that your passion for what you're doing in your work has really come through, and I can see how it's aligning so perfectly with this meeting for you. What else have you got?
I think the excitement right now is using these technologies, and now we have a lot more targeted therapies available. So I think that's one of the themes that I've seen is seeing a lot of single cell RNA sequencing, and I think the spatial platforms are going to be really cool, where you can actually see what's happening on the tissue level. But one other specific talk that I really liked was focused on Tregs and using essentially T regulatory cells that target specific antigens. And there was a talk by Miho Mukai, and she focused on taking Tregs that were specific against the desmoglein 3 autoreactive T cells, putting those Tregs into mice and showing that those Tregs can actually dampen down the inflammation and the blister formation in those mice. So that is really, I think, an exciting segue into what the next 10 years I think will hold, because now that we're identifying specific clones and specific cells that are responsible for disease, ideally we can maybe avoid broad immunosuppression, and maybe in the next 10-15 years we can have approaches that can actually target specific cells that are autoreactive and stop them, and in a very precise way cure disease for more of a long-term thing. So that's I think another thing that I was really excited, because there was a little bit of hint, a little bit of feel of like what's down the line. And
Right.
So I think that was another talk that really for me was inspiring. I mean, among the dozens that I've heard.
Yeah. I really
So-
love that you are getting so much out of this meeting that you're able to really think big picture and think about the next 10 to 15 years and where the research might be going and what those implications are for patients. I think that's exactly what this meeting is about, right? It's about
Absolutely.
breakthroughs in science. And, these are some of the greatest scientific minds in dermatology and immunology and that we have. So, I mean, it's pretty incredible.
Yeah, I mean, it's great to talk to people. I think another thing that's really good about this meeting is you can see what other techniques people are using, and it's almost like cross training, where you're learning from different experts, and you can take those applications and use it in your own field. So I think that's another thing that's really irreplaceable. And then of course, meeting people face to face. A lot of us have been stuck with not being able to travel as much. So now to actually meet people and have spontaneous conversations is extremely valuable. I mean, there's definitely an opportunity to network as well.
I love what you said there about you know, being able to have sort of this cross training, because this is, of course, an international meeting, and we're seeing how other countries and other derm societies
Right.
are doing things. And that is just invaluable, right? Like
Mhm.
sometimes we're bogged down in our own country's red tape, our own country's FDA, blah, blah,
Mhm.
blah. But right now, the doors are wide open, and we get
Right.
to see how these other countries are maneuvering through their own red tape and getting their
Right.
studies done. It's really
Right.
cool.
Yeah, it's amazing how people are able to work together and,
Yep.
and get valuable clinical samples, and it's a lot of work up front, but then you benefit from that work potentially for decades, and of course it helps the patients too. So it's worth the hard work.
Yes, it is. Thank you so much for joining me, Dr. Oldenberg. This was a lovely discussion, and I wish you the best as you continue through the meeting.
Yeah, thank you.